Hello, welcome to ONCOLife interview session. It's my pleasure to introduce our guest today, Dr. Rafael Amado, Head of the Global Research and Development at Zai Lab. Thank you so much for joining us, Dr. Rafael.
Thank you for the invitation.
Could we start with some information about your professional journey and your background?
Sure. So again, my name is Rafael Amado, and I'm the president of research and development at Zai Laboratory. I'm a physician scientist. I trained at UCLA as an oncologist, and I was in academia for 12 years. I then moved to industry, and I've been focused on developing drugs for over 20 years now, trying to get innovative ways to maximize the clinical benefit for patients in diseases that are grievous illnesses, clear unmet needs. And I've had the privilege to participate in the development of drugs that are in the market. I also work on engineered cell therapy. And I joined Zai not just to foment the growth of our regional pipeline in China, which was incredibly effective already, but fundamentally to build a global team that can develop drugs to which we have global rights. And in that space, we have made really great progress, including some of the compounds that you mentioned before that we will be discussing today.
Can you talk to us about the Zai's global research strategy as a whole? What compounds and therapy areas does the Zai prioritize, and what consideration inform this decision making?
Yes, a very good question. Zai was founded as an open innovation company to develop and get drugs approved and commercialized in China, drugs that come from partners that don't have a presence in China. Today, which is over 10 years since its inception, it has a number of products in the market, but it's also a research base as well as commercial stage biopharmaceutical company. It's based both in China and the United States. And the focus is all the way from discovery, development, and commercialization of innovative products. We only focus on unmet medical conditions, and the therapy areas that we focus on are oncology, immunology, neuroscience, and infectious diseases. But by and large, oncology and immunology encompass about 90% of what we do. So our goal is to leverage the competencies and resources that we have to either discover or in-license products to impact human health worldwide. And as I mentioned before, we now have the intent to do this globally as well. Within every therapy area, we have areas of focus and areas of lower interest, but we are also opportunistic. But in oncology, we focus on antibody-drug conjugates. We're moving into T-cell engagers and immunocytokines. And in immunology, we have more of a disease focus on diseases of the skin, the gastrointestinal tract, and in rheumatology. So this is sort of the makeup of the company. We're here to help patients in a space where there's an unmet medical need, and we do it either through internal discovery or through external partnering.
Zai Lab recently presented update data related to ZL-1310 at ASCO. What set this ADC apart from the other in the field? And can you tell us more about what's the next exciting compound?
Yeah, so I'll say more about ADCs, but we are very excited about 1310. It was the first product that we licensed when I arrived. And it's an antibody-drug conjugate that we're developing for second-line small cell lung cancer. And then subsequently, we will develop it in first-line. And also, we're doing studies in neuroendocrine tumors. I think I will talk about the mechanism of action later, but the key features is that this is an incredibly active antibody-drug conjugate. It delivers the payload, which is the chemotherapy, to the target, which is a tumor. And with the right dose, which is 1.6 milligrams per kilogram, the overall response rate is 79%, and 100% of the patients have disease control. So we don't have the durability yet because patients haven't progressed, but there are patients at this dose that have been on study for over 6 months. And this can be achieved with very low treatment-related adverse events. So for instance, the grade 3 rate is only 6%. And there are actually no discontinuations. So we think that this is really the dose that we will choose. In addition to high response rate and good tolerability, the drug is very active in the brain, and in small cell lung cancer, that is very important, including metastases in the brain that have never been treated, where the response rate is in the 80% range. So there are other antibody-drug conjugates in this space for small cell lung cancer with other targets, but we think that we're first in class for DLL3, which is the target of ZL-1310. And for now, we think that this is the most potent ADC in this space. So we're really happy with the prospects of developing this expeditiously in an accelerated fashion in the next couple of years.
Could you also share some more information about ZL-1310, especially about mechanism of action and how it was developed? How do you see its unique design addressing the current gap in the cancer treatment landscape?
Yeah, so antibody-drug conjugates combine antibodies that bind specifically antigens in the surface of the tumors with powerful anti-cancer molecules that are joined to the antibody with a chemical linker. And in the past, the fidelity of these payloads or chemotherapy molecules being linked to the antibody and only decouple in the tumor bed was actually not very well achieved. And as a consequence, there was a lot of circulating chemotherapy that damaged normal tissue. So we have a next generation ADC, which is essentially based on a science called T-mailing, which is a platform that employs a stable linker. So stable linkers are such that can be cleaved in the tumor microenvironment, and the antibody remains intact and it can internalize. And then once it's cleaved, the payload diffuses through the tumor microenvironment. It can enter the cell, it can kill the cell, it can efflux the cell and go to a bystander cell that may not express the antigen, but yet can receive chemotherapy locally and also kill those cells. And in addition, the cleavage of the linker can happen upon internalization of the antibody. So there's really two mechanisms whereby this antibody-drug conjugate can kill cancer cells. And when we look at the pharmacokinetics of the payload, which is really the toxic moiety and the intact antibody, there's about 4 log differences in circulation, meaning that we have a lot more intact drug, which is really not toxic, than we have payload, which is really the moiety that causes toxicity. And this is achieved by having stability of the linker. The stability of the linker is achieved by having specific amino acids to which the linker binds. And also the number of molecules, you can dial them up and down. In our case, there is called DAR of 8, which is the drug-antibody ratio, meaning that there are 8 molecules of chemotherapy per antibody, which is a relatively high number. So in summary, and this field has really gone through a renaissance whereby we now can deliver chemotherapy at the site of the tumor bed. We can do it in the microenvironment. We can do it inside the cell. We can achieve a bystander effect. And these antibodies have great permeability, great avidity to, for instance, as I explained before, penetrate the brain and also exert anti-tumor activity there. And the amount of payload and the amount of chemotherapy that circulates can be minimized, thereby reducing the off-tumor effect that we call, meaning that the rest of the organs are less affected, and the therapeutic window is broader. So our antibody has a very high avidity, and it has all these characteristics that I just mentioned, and perhaps that's the reason why we're seeing response rates in the high 70s.
Bridging the immunology pipeline, could you walk us through the science behind the ZL-1503? How does its dual targeting of the EL13 and EL31R help address both inflammation and itch in atopic dermatitis?
Yeah, this is a very smart design of a bispecific molecule. Of course, there are inhibitors of IL-4, inhibitors of IL-13, inhibitors of IL-31, but this is a bispecific that traps IL-13 in the circulation so that IL-13 cannot bind its receptor. And then it binds the receptor of IL-31. And the ability to bind one does not impede the ability to bind the other. So they can bind simultaneously or they can bind separately. And we just presented this data, preclinical data, at the EAACI Congress in Glasgow. And we demonstrated first of all, that it has a very long half-life. The main indication for this bispecific is atopic dermatitis or eczema. And the problem there is a cycle of itching, scratching, and damage of the lesion. So IL-31 is anti-pruritogenic, so it stops the itching. And then the patient will not need to scratch and thereby damage the eczema lesion. And then IL-13 can come in and allow the lesion to heal. So I think that's the beauty of putting these two together is that the itching stops immediately. And we've modeled this in primates. And a single dose is able to block signaling of IL-13 for about 130 days, which is incredibly long for these molecules. And we challenge the animals with IL-31, which produces a lot of pruritus, a lot of itching. And the animals that received this compound exhibit no, itching phenotype. And again, the longest that we've seen this effect is 130 days. So imagine a patient that is relatively well controlled but has eczema and can every couple of months with an auto injector injects himself or herself and reduce the symptoms immediately and have all these lesions heal. there's still, even with the advent of all these cytokine blockers, there's still an unmet medical need, because some of these, the ones that exist, can cause side effects such as eosinophilic conjunctivitis with IL-4 inhibitors that cause eye disease and other toxicities. So 1331 are better tolerated, and they're active at relatively low doses. So of course, this is all preclinical. We are writing an IND that should be accepted hopefully by the end of the year, and we hope to put the first patient by the end of the year and start observing how it works in atopic dermatitis, and there should be other diseases in which this compound should have activity as well.
You presented compelling preclinical data at EAAC 2025. Could you also share some key finding and explain how the ZL-1503 dual mechanism set it apart from the existing biological treatments?
Yeah, I think it is the introduction of cessation of itching and scratching, which is really very, very morbid for the patient that suffers this. And that immediately allows IL-13 to come to work and heal the lesion. And so we needed to verify this. The mechanism of IL-13 was verified by looking at downstream signaling of IL-13, which is called STAT6. And we saw that the phosphorylation of STAT6, which is the activation, was blocked again for over 130 days. And this is not seen even with an antibody, where after several half-lives, then the potency diminishes. And then the itching is done with an actual model of itching, injecting IL-31. And the key finding here is that these inhibitions of both pathways correlated very well with the PK of the product. So if the PK was longer in some animals, the phenotypic effects of the blockade of these two cytokines was correspondingly longer as well. And then lastly, we dosed these animals up to 150 milligrams per kilogram, which is a really high dose. And we really didn't see any untoward effects. So it was really, really well tolerated. And again, we know that they combine one cytokine, one receptor, or both at the same time. And the potency is really not affected. So all these properties are hard to obtain with a single molecule. So even though these targets are not novel, what is novel is putting them together in a single compound with the right stoichiometry for them to block in the correct position and interrupt the symptom that is most bothersome to patients, and thereby allowing healing of the skin lesion. So we think that by doing that, we will have superior clinical outcomes, as well as ease of use from the patient, and a long half-life that would permit long intermittent use.
Beyond atopic dermatitis, you mentioned potential application in other IL-13 and 31 driven diseases. Could you share which indications Zai Lab is currently evaluating or considering for ZL-1503?
Yes, so atopic dermatitis will be a big indication, because one has to do studies in adults as well as in adolescents or teenagers, and this is also a disease of children. So those are different studies, and one has to do more than one study and have long-term safety. So So even just atopic dermatitis is which is the disease with which we're going to start, is a very large development program, and there are a lot of ancillary studies that need to be done as well. But to answer your question, there are other itching diseases like prurigo nodularis, which is a rare disease that causes tremendous pruritus. And then there are other indications that potentially could lend themselves to treatment with this compound 1503, such as eosinophilic esophagitis, which IL-4 can treat. There's no reason why IL-13 wouldn't be able to treat this. Also nasal polyps, and importantly, asthma. There's something called type 2 asthma that can be inhibited by IL-13. And this obviously would have a very different development path, but also in preclinical models of non-human primates, we have seen reversibility of asthma bouts by the use of this. We haven't published or presented this yet, but we will do so as we complete the studies. So it could potentially be a franchise of multiple indications, and we will have to gauge ourselves, where we play and which diseases we target first, and potentially work with others in the field to try to maximize the benefit of these drugs across all these morbid diseases.
Finally, what role do you see for the ADC particularly in relation to ZL-1310 within your current oncology pipeline? And similarly, how do you see the bispecific antibody like ZL-15 shaping the future of immunodermatology?
Yeah, in terms of ADCs, we have been very active. As I mentioned, our technology, our platform leads to a very stable payload, and that allows us to find the dose relatively quickly. And we follow Project Optimus from FDA to try to get the minimum dose that has the maximum effect, a minimum toxicity. We think that with 1310 we are there or very close, and we're working with FDA on that. But in addition to that, we have other targets like LRRC15. We haven't talked about that. That is in sarcomas and mesenchymal cells. That's also going to be an IND this year. And it could be given because the target is in cancer cells or because the target is in malignant fibroblasts in the tumor microenvironment. So ADCs have come a long way, and there's actually an evolution of them. For instance, there are ADCs that target more than one epitope in the same target, or target different targets and different epitopes. Those are called biparatopic. And those are likely to be more effective, and the delivery would improve. I think the permeability will improve. And importantly, ADCs have been very successfully combined with checkpoint inhibitors. Because they cause immunogenic cell death, it attracts T cells that can come in and then recognize not just the canonical antigen that the ADC recognizes, but other antigens that get liberated from the dying cell. And because the checkpoint inhibitor removes the brake from those T cells, then you get orthogonal killing by the ADC and by the immune system. And this has been shown with ADCs that have completely supplanted chemotherapy. and have been combined with checkpoint inhibitors, like for instance PAD7 in bladder cancer. So as we move into phase 1, we will attempt to remove chemotherapy from the treatment of small cell lung cancer and continue the checkpoint inhibitor that is a standard of care in small cell lung cancer. So we will continue to develop ADCs and we will continue to advance this field. The T-cell engagers are work in a completely different way. They recognize, they are bispecific molecules that recognize a particular antigen. For instance, tarlatamab recognizes DLL3 as well. And on the other end, they recognize CD3, thereby activating T cells at the site of the tumor. Again, they're orthogonal mechanisms of action and really very worthy to combine both ADCs and T-cell engagers. And there's been news about Imdeltra prolonging survival. And we plan to develop a clinical trial that will start this year in second line, comparing 1310 to standard chemotherapy, including Imdeltra. With regards to the bispecific that I mentioned before, that may have a lot of applications. I think it's a really good molecule for the reasons that I specified. And the effect is immediate and very long-lived. So if we can recapitulate those characteristics in patients, as we are doing in the preclinical setting, then I think this can be a really important drug for patients living with these grievous illnesses such as atopic dermatitis and other diseases as asthma, other pruritic disorders, and alike. So we're very happy with this initial foray into global drug development. We have other molecules that we don't have time to talk about, but we would like to get at least 1 or 2 INDs every year and move, every couple of years or so molecules into phase 3 studies. And the first one will be DLL3, 1310, and we hope that we can get approval, accelerated approval in as early as 2027, and that would be really gratifying for us, to have our first global molecule, be at Zai. It will just give us a global projection, which is really what we intend to do for patients. So very excited about building a world-class team, and very excited about the fact that many of these molecules came from our internal labs. So we have a discovery group that has been delivering, and we intend to take those molecules as far as they can go.
Thank you very much to be with us today, Dr. Rafael Amado.
Thank you. It was my pleasure. Thanks very much for your questions.